So, welcome everybody to today's webinar, which deals with many topics from API to quality control, precise milling and sieving technologies for pharmaceutical application. My name is Dr. Tanja Butt, and I'm working since more than 12 years at RETSCH as product manager and also in marketing and application support. And I hope to give you a good overview about different topics I'd like to cover here. Many of these topics require at least 1 hour webinar by themselves, like nanogrinding or sieving or whatever. So this is just an overview here, and I hope makes you curious enough to go to our web pages, to contact us later on, or to watch the other webinars on demand, to give you a good impression what is possible. I hope you can hear me clearly in this webinar, and you can also ask us your questions. If you type in a question in the chat room, we can see this and answer it later on or during the webinar. Depends on how many questions arrive at the same time, but you will get your answer for sure. If the question is of, let's say, more general interest, then we will refer to it at the end of this webinar as well, for all the participants. And you will also receive a download package with the follow-up mail after the webinar. So you get the recorded session, the complete material like the presentation, also videos, applications, reports, and so on. So please make use of it, download the download package, and then you have a lot of information in your hand to, which maybe help you in your daily lab work. So let's start with more or less quality control reasons. The milling task. Why do we have to homogenize? Why do we have to do the milling steps before we can analyze something? The grinding serves 2 topics. We need to do the homogenization steps in order to assure that we get reproducible results, that the sample we analyze is always the same. Yeah, and we also need to assure that the sample we analyze in the lab represents the original sample. Yeah, that it is not only reproducible but also representative. And this is very important, because if you do not pay attention here and we do something with our sample, then we can use the analytical method we want, or different methods like HPLC or XRF or gas chromatography. Everything is there. Most of the times the analytical method is expensive, the device is expensive, but if you do any mistakes prior to the analytical step, then you cannot compensate for this anymore. So it's extremely important that you pay enough attention to the step before the analytics in order to get really reliable results in the end, which are really representative and reproducible. Yeah, and this is where RETSCH helps. Let's see just an example like this plastic bricks, Lego bricks. Maybe not pharmaceutical application, but I think it's clearly visible what is meant. So, for example, you can only just pick one of the bricks to analyze it, because the analytical device only takes this amount of sample. And in one of the different colors, maybe toxic substances. So if you can pick only one and you pick the wrong one, maybe only the red, you won't see the substances and you will claim it's not toxic. So you have to homogenize it in order to get a little bit of the blue, of the yellow, and of the other colors in the few milligram of sample you can analyze. Yeah. So here maybe in this intermediate, you see, okay, now I have every color present, so I should see the toxics. Yes, you see them, but is it the right? Is it really quantified? And do you really see the true amount of the toxins? Because maybe there's only one yellow part, and this is underrepresented So if this is a toxic part here, maybe you do not really see the true value. If you just then homogenize it fully, you have the same composition as in the original sample, and you always get the same result, whether you pick the sample here, here, or here. Yeah, so it's reproducible and it is representative. You really get reliable results in the end. Application example, pizza. If you do it only coarsely, like in the orange, yeah, and then you analyze 5 times the fat content, you see that you have variations, because in this sample here, you picked one with a large, let's say, cheese remaining with a lot of fat. If you fully homogenize it, like in the green, you see that it is more stable. Yeah, you do not have the peaks, but also not this underrepresentative samples. So here, that is a true composition. You get reliable results only in the fully homogenized one. So here, yeah, I think these are 2 examples why improper sample preparation can really influence the results. And of course, especially in pharmaceutical application, you need to avoid things like that. Yeah. So you need to assure that the sample preparation is really adequate to the pharmaceutical requirements, let's say. Let's come to the fineness. A lot of customers tell us, I grind as much as possible and as fine as possible. Yeah, you can do this, of course, but it takes time. The finer you like to get, the more time you need to pay. So when it is not required, why waste time? And for most analytics, an intermediate particle size is fine. So let's say 0.5 millimeter particle size is really good for most analytical instruments. There are exceptions. XRF, for example, needs finer particles. There you have to pay a little bit more attention to come to finer particles. Nanogrinding, for example, as well, for not only analytics, but for better solubility and the products is also very important. But for quality control in most steps, you do not need such fine particles. So really important, not as fine as possible, as fine as needed. Yeah, only make sure that the sample is homogenized and that you achieve at least the 500 microns. This should be okay for most. And when we talk about fineness, just this picture over here, before and afterwards, you see here the D50 value is below 3 millimeter before and below 40 microns after the grinding process. We also talk many times about the D90, means 90% of the sample is below a certain value, or not 50% of the sample is below a certain value. This is also a nice, let's say, result you get after sieving processes. We see this later on in the presentation when we talk about sieving. Okay. So in total, what we can do and what we can offer, we can play around with different grinding principles, different mills, different preparation steps, and different process parameters. Yeah. So here we have a lot of option, a lot of combination, which can be used to come to a homogenized sample in the end. And this is not only facts, it's also experience. For example, how the sample material behave, what is maybe the best mill for it, or the best grinding parameter, speed, amount of balls I use, and so on. So this is why we call it the art of milling. Yeah, it's not only theory and logic, it's also experience. And we are hopefully there for you to help you through all these different options to find the right solution, yeah, for your sample tasks. And when we're talking about grinding principles, here we have 5 different grinding principles. You see this, and some are more suitable for the hard sample materials and more for the soft sample materials. Pressure, yeah, for stones or brittle materials. We have impact, like glass bottle thrown on a wall. This is one impact event. Of course, for hard sample materials, very nice, but for a bundle of plant materials, there won't happen anything, so not suitable. We have friction, which is intermediate, hard sample material, soft sample material. This is also clear. And we have for the soft material, we have shearing, yeah, like for cheese, you know this from your kitchen, or cutting effects. Yeah, you just cut fibrous material, softer sample material, elastic sample material. You cannot cut a stone, on the contrary. So here, depending on your sample properties, hardness, brittleness, softness, stickiness, and so on, you need to adapt the different grinding principles. You cannot use everything for all materials, and this is also a reason why we have so many different mills on the market, because they combine different grinding principles in different ways in order to cope with all the different solids in the world which needs to be homogenized. Yeah, and this can be seen here. For example, jaw crushers and vibratory disc mill, hard or medium hard samples, quite nice, but not suitable for soft ones. Yeah, so with the pressure, you cannot just crush, let's say, some plant materials over here, yeah, or some tissue materials. Rotor mills, cutting mills, knife mills, more for medium hard to soft materials, and mortar grinder and ball mills suitable for the complete range. So it needs really to be seen what are sample materials to come to the right machine, and also, yeah, is it elastic, is it sticky, and so on. Needs to be taken into account. Yeah. The mills, we also can use different grinding tools. This means, for example, rotor mills, we use different rotors. For ball mills, we use different grinding jaws and balls. And for the jaw crushers, we use different breaking jaws. And they can be made of different materials, yeah? Different steel types, tungsten carbide, zirconium oxide, silicon nitride, PTFE, or agate, depending on about what we are talking here. And you see that these different materials of the tools we are using, they have different hardnesses and densities. And one rule of thumb is that the material we need to grind needs to be softer than the material we use for the grinding. Yeah. So if you use PTFE as a grinding material and you have a very hard material, sample material, this does not work. Also with the agate, maybe this does not work. And here we have always pay attention that our tools are harder than our samples. Density plays more or less a larger role in grinding jars and grinding balls. So the more dense the sample the tool material is, for example, the balls, then you bring in more energy into this grinding process. Tungsten carbide is a very dense material, very heavy material. So a ball of 20 millimeter size can accelerate or bring in more energy and input at a certain speed of the mill than, for example, steel balls of the same size. And then you have a greater crushing efficiency with the tungsten carbide balls. But on the other hand, most of the crushing energy is maybe not required for softer materials. Then you only get sticking of the material. So maybe here stainless steel is better. So you can influence the way how the sample is crushed with the choice of the right material. Yeah, and abrasion is a very important factor for pharmaceutical applications. Tungsten carbide, zirconium oxide, very abrasion resistant materials. PTFE, you can scratch it with fingernails, so it's a contrary of that. So you need to take all these different factors into account to choose the right material for the grinding step. And here, very important, every grinding operation causes abrasion. You cannot avoid it. If you work with stainless steel, you have some stainless steel abrasion. If you analyze for heavy metals or some iron in the sample, then maybe this is not the best material. You should go to zirconium oxide instead. Yeah, this can be also seen in this slide over here, where we ground glass. One time zirconium oxide tools, and the second one and the red one steel tools, and you see that it is a grayish color. Yeah, you always also see this discoloration. And when we analyze this with XRF, you see with the zirconium oxide, there aren't any peaks for iron. But for steel, when we use the steel equipment, we see some abrasion of the steel. It's not maybe an broken steel parts when you did everything correctly, but a certain amount is there. And this is very important. The material properties being analyzed, of course, they should not be changed. You can find a workaround, like using zirconium oxide when you are interested in heavy metals, for example. Yeah. Then I'd like to come to quality control aspects. And here I'd like to point out rotor mills like ZM 300 or SR 300, cutting mills or ball mills like MM 400 or PM 100. And I need to switch off the laser pointer in order to get it started, I think. So here, ultracentrifugal mills, you can use the DR 100, which feeds the material into the machine. This is suitable not for stones, but for softer, medium hard materials like grain or lactose powders or plant materials. And as soon as the sample hits the rotor, which rotates very, very fast, it is accelerated towards this teeth, and then you have a single crushing event, and then the pre-crushed sample comes between the ring sieve and the juice of the rotor, and it is further crushed by friction, and you then get the very fine particles in the collecting receptacle as soon as they fit through the apertures of the sieves. By changing the sieve aperture sizes, you can influence, of course, how large the sample is in the end. Yeah, larger aperture size, larger particle, finer aperture size, finer particles. Then let's come to a quick look for cutting mills like SM300. Here we again work with the rotor. When the sample comes to the rotor, now we see it from inside. We do not work with teeth anymore, but with really blades cutting. Yeah, and we have also the cutting bars in the housing of the machine, so that the sample are really cut, and as soon as they are fine enough to fit through the bottom sieve or the apertures of the bottom sieve, they are collected. And again, you can influence the size, but the size of the aperture size of the sieve. And a ball mill, we use balls and grinding jars like this. In this case, this is an MM400, where you can crush a maximum sample quantity of 20 milliliter in one jar, or smaller, depending on the jar size. We usually use one large grinding ball, close it, everything, put it into the mill. Very simple process, quickly done. And then you see that we have a horizontal movement of the jar, which brings in a lot of impact to the sample, a lot of energy input as well, depending on the model. And you see that this large ball now crushes the material inside the mill. Yeah, but this horizontal movement mostly impacts. Okay, so I think I will go on. So here we have 4 or 5 different dragees and pills. We have many, many more crushed in our lab for test grinding. And this is a nice example for the sentence, a pill is not a pill, yeah, or a wood sample is not like the other wood sample, or herbs are not always the same, yeah, can be very different, and even pill have different properties. The size of the pills, for example, can vary from here to here, or we have coated tablets, maybe they are also liquid inside. They are sticky with the coating, like the sticky dragees and so on. So depending on the sample size, the sample amount, and the stickiness, we come to, the choice of the right mill, I think. So here are a lot of details, which equipment is used and so on. Remember, you will get the presentation later on, so I will not get to each detail here. But what I like to show is that, for example, larger quantities maybe need the SR300, which keeps larger quantities. Only 7, 10, 15 gram can be done in the ZM300. We can influence the result with the speed and also with the bottom sieves or ring sieves we use, yeah, and can get very, very fine powders in the end with the ZM300. And here, special example, the MM400 with this coated sticky dragees. You see that we embrittled it with liquid nitrogen before. This means we take the grinding jar, the sample into it, the ball into it, close everything, and then we immerse it in liquid nitrogen. Why? We need to get rid of the stickiness, and we can only do it by embrittling it. And while it is immersed in liquid nitrogen until it stops bubbling, you can take it out and put it into the machine. Then it's pretty cold inside and really embrittled, and the stickiness does not play any role anymore. You can pulverize it. This would not be possible at room temperature in this case. Also when it is liquid inside, yeah, the dragees. So here, many details to be covered, yeah, in the terms of equipment. Just examples on what different types we can use and what can be the results. The same for plant materials, which also play a role in pharmaceuticals, yeah, for TCM or whatever you need. Dried roots, dried stem parts, dried fruits can be easily ground in the cutting mills or in the ultracentrifugal mill ZM300. And you get here more or less, you see, not that fine particles, 1 millimeter. It's possible to come a little bit further down. It might be a problem, as in those two pictures, as you see here, that it is fibrous material. It breaks as fibers. Yeah, and it is not that easy to get rid of the fibrous part because they can pass lengthwise through the bottom sieve or the ring sieves. And if it is really important for you to come to more round shaped particles, then a ball mill needs to be used in a second grinding step to get rid of this, let's say fibers. Yeah. Same for different roots, herbs and spices, or even orange peel like this. Okay, and also very nice for some of you maybe, implant materials, pig teeth, human bones, bone cement, and so on. Very hard material can also be crushed in cutting mills. But here we need to change the rotor, not a blade anymore, because blade and pig teeth, this won't happen. But if you change to a more robust 6-disc rotor, then the same mill is able to crush even this hard materials. So what I want to tell you here is with the same mill, but choosing different equipment, you can adapt it to many different sample materials as a rule of thumb. And this is also true for all the other machines where we have different equipment. You can adapt it to your specific requirements. Here again, 6-disc rotor, and here again, embrittlement for this more sticky sample material. Here we have also used the cryomill for the fine grinding step. Yeah, this is a pre-grinding step in the SM300, then the cryomill, same material in the cryomill. And here we use really brittlement at almost -200 degrees in order to keep it pretty cold and brittle it during all the grinding time. And then we can also pulverize such sample materials. Okay, coming away from just this grinding purposes, I think that also mixing purposes are very important in pharmaceutical applications. And here I'd like to quickly show the working principle of the planetary ball mill system. Here we have again different models, 1 to 4 grinding stations, and you see that it is now a circular movement, not a linear movement as in the MM400 anymore, but you're working still with the jar and many balls in this case. Here you get more friction, also a little bit of impact, of course, and this mill is very efficient also to crush sample materials, to homogenize them, but also to mix them. And we have larger grinding jars, so up to 220 milliliter sample per jar can be crushed or mixed. And how efficient the mixing is can be seen in this picture over here. When we mix just pigments in different colors, we get after only 5 minutes in the PM400, this is the one here with the 4 grinding stations, we get a homogeneous mixed result. And this is also possible for larger sample quantities using our drum mill TM300, where the largest grinding container is for mixing purposes 21.7 liters. So here much sample can be mixed at the same time. Then we need to talk about temperature control, very, very important for pharmaceutical application. And we need to know that each grinding operation generates heat. We cannot avoid it. It depends on the mill, on the configuration of my sample, on the sample volume, the jars I use, for example, the sample quantity, how warm it is. So many factors influence the temperature, but we cannot completely avoid it. What we can do, we can control it. Yeah. So we have different mills which allow a kind of temperature control, at least measurement, observation of the temperature, so that we have at least a picture how warm our sample was in the grinding process. So when we're talking about the Emax, which is a ball mill, we have cooling options. You see this later on in some slides, and we have also temperature control mode. So you can tell this machine, okay, stop grinding when the sample gets, or when the jar is getting a temperature of 80 degrees, for example. Then it stops grinding and it starts grinding with a lower limit, maybe 60 degrees when you tell it. And it starts grinding again. So it only grinds between 60 and 80 degrees. So you can be sure that it is not getting warmer than this, and that not too much time is given away with too long cooling breaks in between, for example. With the ZM300, which we have seen already in action, we have at least an indicative temperature in the display, so you can watch and observe how warm the sample is getting and stop if it is too warm. I already told you about the cryomill, where we have continuous grinding at almost minus 200 degrees. So the complete jar is flushed here with liquid nitrogen inside. This means very, very harsh condition. You can use it for crushing every plastic material, but it's very cold as well. Yeah? So here, for example, when you use this, volatile ingredients like terpenes are preserved. Yeah, they stay in the sample, no chance to evade. And we have the MM500 control. And here, this is the only mill which can heat and cool both from minus 100 to plus 100 degrees. You will see later on why this heating is maybe also beneficial in terms of quality control, just crushing. We are mostly talking about the cooling options. When we're talking about this machine, I'd also like to show you how it works. So here you see that the jar is placed on the thermal plates with a flat surface, and the thermal plates, they are flushed with the liquid. This can be liquid nitrogen, then you have really good cooling capacities, heat is dissipated, or you can use a thermal oil for heating purposes, which is pumped through the system, and then you bring heat to the jar. Yeah, for example, for mechanochemistry. Later on, I will show this. So here we have many options to work with liquid nitrogen as cooling agent, or to put a chiller on it and work with minus 20 degree cold water glycerol mixture, or 4 degree cold water, or a thermal oil for heating. Very, very nice and flexible system. And when we are talking about warming effects, the natural warming effects, you can see this in this slide. Here we took a glass sample and opened the jar to measure the temperature of the sample inside. The machine gives you the temperature of the thermal plates, but this is not the real temperature inside. Of course, after a while, it adapts to each other, but there's a certain delay. And if you're really interested in the sample temperature, then you have to open it and to measure it directly. And we did this in this case, and what you can see when we start at room temperature, no cooling, just as warm as it is, and let it run. In this configuration with the 20 millimeter bolts, you end up, let's say here around 60 degrees with a 125 milliliter jar, and lower with a 50 milliliter jar. So again, how you can influence the temperature, yeah? Choosing different jar sizes also has an influence. So no cooling, and you end up somewhere between, let's say, 35 and 60. If you set the same machine to cooling, please keep the temperature of the thermal plates at 0 degree. When you use liquid nitrogen as a cooling agent, you see that I can influence it drastically and it stays around 15 degree, yeah, the sample inside. If I put it to the harshest conditions, please stay at -100 degree at the thermal plates and regulate the temperature of the thermal plates at -100 degree. You see that the temperature inside is at around -80 degree. And when you then also set some cooling breaks, yeah, which means you start grinding, stop grinding after a while, let it cool down, start grinding, stop grinding, and so on. So in these iterations, you can keep the sample inside very, very cold, close to the -100. And this is the way you can also grind licorice, for example, yeah, or any dragees with liquid nitrate, with liquid filling, or any plastics. Yeah, this is very nice. You use, in this case, the M500 control and with the 125 milliliter grinding jar, the cryo pad, some of the larger balls. You have to pre-cool it at a certain time, -60 degrees set at the thermal plates, and we grind here also in this grinding and cooling intermediates breaks, and after just some minutes we have it pulverized. And this is true for a lot of samples: herbs, plant materials, pharmaceuticals, cellulose, elastomers, polymers, biological samples, and so on. So to sum it up, the MM500 control covers a very large temperature range where you can control it. Contrast to ball mills without any active cooling or temperature control, then you start at maybe room temperature, but it heats up depending on what you set up, which equipment you really use. It can be really warm. The Emax with the 4 degree cold water cooling can countercool it a little bit, so you do not end up at the highest temperatures. And the cryomill stays around -200 degree. Yeah. And when we're talking about the Emax, I'd also like to show you here the grinding principle of the Emax, which is a bit different to mixer mills or planetary ball mills. It combines both of the worlds, and it has an enormous speed of 2000 RPM, up to 2000 RPM. And what you can see here with the movement, it's not horizontal and it's not circular, it's both. Yeah, it is a best combination of mixer mills and planetary ball mills. You see that the jars are shaped oval, but they make a circular movement. And this leads to very, very high mixing effects. And you get at the same time friction and impact events. Yeah, mixer mill is more impact, planetary ball mill is more friction based, depending on the ball size and the filling degree. But here you have same at the, or both effect at the same time, and this leads to very, very high capacity to crush samples, and also the 2000 RPM, of course. And the 2000 RPM as a maximum require cooling. Yeah, so we need an efficient water cooling, which can be seen here on this picture. We have here this water reservoir, and then the water is pumped through the Emax and through the jar holding system and cools the jar. And this internal water cooling system can be countercooled with a chiller, so you can here dissipate the heat. And of course, we have the temperature control mode with a set maximum of what I told you, 80 degree, for example. Yeah, so that the sample material is not overheating. And nanoparticles, they really need time. Just have a closer look at this point here. PM3 not at 650 RPM is the blue line, and with an increased speed at 800 RPM is the orange line. And when we have just the fineness measured over the time, the net grinding time, not any pauses or breaks in account, then we see here that you need approximately 2 hours with the lower speed, and you reach the same fineness at 30 minutes with the higher speed. So here the speed really has a very high impact, but it needs at least 30 minutes to hours to achieve almost nanoparticles. In this case, it was 200 nanometers. Now, for this part, not every material can be ground to nanometer, or even with a lot of effort and a lot of grinding time, it's very difficult. 200 microns, okay, but then it's really getting difficult. It depends strongly on the sample if it is possible or not. Save time. This is how you can do it. You can select the right machine for it, because some machines heat up more than the others. And this depends on the design of the machine and on the grinding mechanism. So mixer mills do not heat up as much as planetary ball mills, and the Emacs can be countercooled. And even in the bench of planetary ball mills and different models on the market, it depends strongly on design if there's a good cooling capacity for the jars or not. Yeah, with air cooling, or if the heat can go away somewhere or not. And when we have benchmark planetary ball mill with highest speed, we ground titanium dioxide as well here, and we achieved 120 nanometers. It took 120 minutes, just net grinding time. But because this mill has not the best heat dissipation, you need a long break to not overheat the sample and the machine. So in total, it makes 600 minutes. Yeah, when you take the breaks into account. When you work with the mixer mill MM500 Nano, then it does not get as warm. You can just take the net grinding time as the total process time, because you do not need any breaks in order not to overheat it over 80 degrees in this case, and you reach 90 nanometer particles. And the Emacs at 2000 RPM only took 30 minutes, and no breaks were required because of the cooling capacity with the 4-degree cold water. So the total process time was still 30 minutes, and we had the finest particles here because of the enormous energy input and the grinding mechanism with the good mixing effects and so on. So here, with the right choice, you can influence how to save time, how to avoid breaks, and also with the Emacs, it is very nicely shown. You get a very narrow particle size distribution, which I think is also important for some of you. And if you're interested a little bit more, details there are other webinars dealing with this topic for very deep details. I just want to show one more slide here for the wet grinding of titanium dioxide. Why wet grinding is required? Why can't I do it in dry grinding mode? Because I need to separate the particles. Yeah, so I need to avoid the agglomeration effects by some surfaces. Loads, and here I can't do it in the dry mode when I want to achieve, let's say, less than 200, 300 nanometer particles. Yeah, of course. So when I use PM200, 650 RPM, again with the very fine grinding balls, 0.1 millimeter. They look like a powder by themselves. Some people tell me, this is grinding balls. It looks like the result after grinding. No, this is grinding balls. You achieve about, let's say, in this configuration, 120 degrees after 30 minutes. When you use the Emacs, it does not get that warm. It's about 80 degrees. And the MM500 control at 30 Hertz is even achieving lower temperature. So here you see the different mechanism of the different machines and how it contributes to the heating up. In contrast to that, the Emacs give the best and fastest, finest particles, then followed by the MM500 control, and then by the PM. Now, here. In the end, you achieve more or less the same result, but it takes longer time in a planetary ball mill or in a mixer mill, contrast to the Emacs. And you have in the Emacs, again, this cooling capacity with a 4-degree cold water to bring the temperature down, the same as in the MM500 control, when you use 4-degree cold water. So here you have many ways to influence the heat. Then let's come to mechanochemistry, completely different. So you see again this picture with a different grinding principle. What is mechanochemistry? Mechanochemistry is a nice way to, let's say, come away from traditional principles of doing chemistry. So a reaction from adducts A and B form product C or C and D. You can do this in a traditional way. You know this. Maybe you put the sample materials you like to react with each other in any solution in a Erlenmeyer flask and heat it up in order to let the reaction run through. Here in this case, we need to overcome this activation energy. You remember this, or you know it much better than I do. And then the reaction takes place. So you need to have a kind of energy source to overcome this burden here. And this is, in traditional, the heating. Here we use impact, pressure, and friction in a ball mill. Yeah. So with all the different grinding principles, you can also do, and this mixture, mechanochemistry. Yeah, and why does this work? There are a lot of scientists dealing with this and trying to figure out how it really works. There are different theories, but what is helping to drive the reaction here is that you get fresh surfaces over the time. Yeah, when you grind particle, fresh surface is created, which can react with the partner. You have also this heating. Now we like to heat it up because you bring in energy input. Yeah, it's contrast to the quality control slides I previously told you to get rid of the heat. Now we want to heat in most cases because it helps to accelerate the reaction or to let it run or to increase the yield. We have a good mixing, so the particles meet each other. Maybe different than in solution where they do not really meet. Yeah, so they are not in this close contact in solution than in this dry mode in a mixer mill or in planetary ball mill. So here, the ball milling is used to come from the adducts A and B to new material. This is very beneficial. You do not use any solvents. You do not have to figure out which solvent. You do not have to get rid of the solvent afterwards in purification steps. It's safe in handling. It's faster sometimes. You don't need hours than in solution processes. Sometimes it even works in some minutes in ball milling processes. You have a nice route to change maybe the reaction pathway. So you get different material from A and B than in traditional way of doing chemistry. So you can form new products you do not get with traditional ones, and you get maybe increased yields. So this is really, really nice way to influence the R&D on APIs. And here is also a visible example with a Knoevenagel reaction, vanillin and barbituric acid, where you see in the beginning it's more or less colorless of white, then it's in the end yellow powder. So something really happens here. Yeah. Examples of mechanochemistry. MM400 was used, and it was used for transform cellulose to cellulose fibers with highly charged cellulose fibers, and they have better performance in order to catch maybe pathogens or filter out something somewhere. Also maybe to catch some APIs where they are not required in water or whatever. So here you can transform standard cellulose to, let's say active cellulose with this mechanochemistry. You can also use the MM500 control in order to take the cooling capacity of it, and in order to not change the reaction pathway, but to stop at a certain point. So at room temperature, reaction goes over certain steps to the end product. And when you set the temperature at -5, -10 degrees, you can stop at derivate 1. Yeah? You do not get the end product as in traditional way or at room temperature, but you can stabilize the intermediate. And this was an, I think, MOF example, where you have this high porous intermediate, and you can really, use it afterwards. On the contrast, heating influences the products. So you change the way of, or sometimes you can change the way of what happens in the end. So it's not product A, it's product B what comes out. And you can see this here. I have no clue what it is, please. There's a publication for it with our collaboration partner, but you see with room temperature in contrast to heating up to 80 degrees, something else comes out. So you influence the product. Or with the heating, you can influence the yield of a MOF. No heating, you get approximately 60 degree or 60% of yield, and by heating it up to 60 degree, you get almost 100% yield. Yeah, so you can influence the yield or the pathway, or sometimes both, with heating up. All the references are on our Mechanochemistry landing page. So they derive from our collaboration partners, and we are allowed to show them, to use them. And there are many more on this page. Please inform yourself if you are interested and just call us. We can hopefully help you. And one more word for mechanochemistry. You can also observe the reaction while it runs. For example, with in situ Raman spectroscopy. Well, you place the Raman probe underneath this jar, which is transparent, so that the Raman can come through it, and the signal also can be detected. And what is very nicely that you can go through this bottom plate of the MM400, so you can unscrew the bottom plate, and then there are two openings, and you can point it very close to the jar, and from the bottom of the jar where always is some sample, yeah, because at the bottom of the jar there's always a sample, and you can focus it more easily than pointing from the side. And there you can really observe the reaction while it's running. And this jars can also be used for light-induced reactions. Transparent light can come in. Light is an energy source, like heating, like the mechanical forces, and you can also influence the pathway and the yield with the light induction and of the PMMA jars. Okay, then let's come to some co-crystallization, but this similar to mechanochemistry, but it does not alter the molecules. It just alters the structure of the crystals. So here we like to use an API and put it or mix it with a co-former in order to get a different crystal structure and alter maybe the solubility of some sample materials or some APIs. It's very interesting. You maybe do not know what is the best co-former, A, B, C, and in which ratio, 1:1, 1:2, 1:3. You need to figure out, so you do screening effects, and you need many samples, small volumes, because you do not waste so much material. You only work in small portions, and disposable vials is also nice. You do not have to clean everything. So here we can offer a special adapter for our planetary Balmer system, which works with this gas chromatography glass vials. Yeah, then you can do nicely the screening. I just want to show you quickly the video, not in full length, how you can do it, and skip the tone. Here we can also use 20 milliliter bottles. Yeah, here's the one for the small one, here the one for the large one. Jars in the background. You mix it with some 3 millimeter steel balls, add liquid. This is very important for the co-crystallization. And then you can also use the safety closure here. So in the unlikely case that one of the glass bottles breaks, which at the right condition does not do, but in the unlikely case it does, so the sample stays into the jar. Yeah, it is equipped with safety closure device. And then you can set it into a planetary Balmer, in this case PM300. Let it run at moderate speed, yeah, no full speed, in order to do the crystallization. And you can also do it in MM400 or in mixer mills. You can use here the steel tubes, small ones, in order to get the crystallization done. And we have also on our web pages, in this case, the pharma landing page, you see all the references that you get similar results in all the different tubes. So it does not depend on the position of the tube in the mill. Yeah, you get the same crystallization effects in each of this tube. So it is really comparable and can be used as well for the screening purposes. And once you are ready with the screening, you like maybe to upscale it to a kilogram scale. You can use our drum mill TM300 for it. And this is also done and published, that you can do the ibuprofen nicotinamide co-crystal here. And this is a very nice example which also shows that you need some liquid. Yeah, not liquid grinding, but some droplets of liquid into it. Not wet grinding as for nano grinding, but it's so-called lac. When you don't add any liquid, it takes time, yeah, to come to full conversion. But if you add some droplets, they get full conversion after, yeah, just 2 hours, something like that. And 3.2 kg scale in this case. And also the abrasion, we talked about abrasion factors. You use steel balls, you have a little abrasion of this, but in this paper it is also published that the steel abrasion is neglectable. It's fine, yeah. And there are many applications reported already, which work in planetary ball mills, but also there are papers more coming out for TM300 at different co-crystals here for upscaling. Okay. Then, before we skip to sieving, some more words about cell disruption, biological samples, and tissues. Here, again, we have a lot of different options, and usually the MM400 is used with disposable vials, Eppendorf tubes, or Falcon tubes. This is just an overview. You can do from plant samples to cells, single cells, and I'd like to cope with the cell disruption of single cells and for tissue homogenization in this overview. For biological samples, we use bead beating methods with glass beads. This is for fungi, bacteria, macroalgae, yeasts as well. Yeah, so such samples can be crushed easily in cell disruption purposes, and homogenization of samples like tumor tissues. So here again, I can talk about 1 hour about the different methods, how to do it, what to think about and how to use it. But I think it's just an idea that for all of you who are involved in RNA, DNA, proteins, pathogens, and so on, this could be also a nice instrument for you. For RNA, DNA isolation, we need small volumes. You know, with PCR reaction, we can multiply it. That's no problem. With proteins, maybe it's more difficult to multiply them, so we need some larger sample portions. And of course, we like single-use vials, not to have cross-contamination and to have this cleaning efforts. So here we offer different adapters for Eppendorf tubes, Falcon tubes, and so on, and different mill types, mostly mixer mills. And here just a picture of algae before and after the cell disruption. In this case, it was 20 seconds of this algae. If you have yeast, it takes a little bit more time, maybe up to 7 minutes, but you come to the same result in the end. And compared to using a vortex, which is commonly used in many biological samples, I know this from my studying time, you have an automated system of the shaking. Yeah, so it gives you better yield and less variations in, for example, the protein concentration afterwards, and it is also keeping it cooler. And when you use them in 500 control, I talked about the cooling capacities. You can keep the vials really cold, less than 10 degrees or even less than 5 degrees. So no protein is degradation is visible here. Yeah, so very nice for proteins when you have this cooling capacity as well. Homogenization of tissue, lung, liver, whatever, can be some kind of tissue from somewhere of the body, which needs to be homogenized. A kind of starter protocol here. Fill up 30% of the tube, whether the small or large one, with the tissue, add the balls, and then fill almost up to the upper end with buffer, and then let it shake at 25, maximum 30 hertz. For example, I'm 400, but maybe also in the other ones, do not go more than the 80 hertz because you have a risk to break the wire. when you have put too much impact into it. That's also the reason why we have 4 times of the large balls, because they hinder themselves to have too much crushing efficiency than one, which can have more impact in the tube. And here, what I like to show you, you can see there are some unhomogenized particles left in this procedure. So there's a risk that it is not fully homogenized. You can only get rid of this when you do it cryogenically. But if you work cryogenically in plastic tubes, you also embrittle the plastic tubes. And then, of course, you have, again, a high risk of breaking the tubes. And you do not like to have virus-loaded tissue spread in your mill. So the solution, if it, it shall not look like this, you need to work, again, with steel tubes. So we have here the 2 milliliter steel tubes, 5 milliliter steel tubes for the mixer mills, and these are safe to use. So you can homogenize fully also this tissues. And one last aspect for biological cells is washing of intact cells in order to see what is involved. For example, which bacteria in this special case are involved in the infection. So you take a little bit of the biopsy material from this person and put it into a wide mouth bottle, add buffer glass beads, and then slightly mix it, not fully, in order to wash off the bacteria from the material. And then you can plate the bacteria and determine which ones they are. And then you have a kind of personalized diagnosis for perfect therapeutic measures. So also this is possible with the help of the MM400. And if you're interested in all these ball milling aspects, you can screen this later on in the home package and come to this recorded presentation of ball milling, with all the details, and of course on our web pages. Okay, then the last slides, I'd also like to tell you something about sieving. Sieving is used for determining the particle size after crushing or in production. Or a quality control inspection of raw products you are maybe receiving. This can be done for a sieve cut, like 90% of the sample is smaller or larger than a specific size. Or you can also do particle size determination, like in the picture I showed you in the beginning, with this curve of the particle size distribution, if all is in the expected range. And we have 11 different machines. Sieving machines, and they are based on 4 different modes of sieving. I think most common used is here this 3D vibration sieving mode, where the particles are moved in a 3D way, and they are accelerated also upwards and downwards and can reorientate and pass through the sieve. Very, very easy process. And for fine powders, especially in pharmaceuticals, also the air-jet sieving is very commonly used, where this nozzle here blows some airstream through the mesh of the sieve and deagglomerates particles, and then when they are fine enough to pass the sieve, they are plugged in the vacuum cleaner or in a bottle when you use a cyclone. So again, for sieving, I can give you a talk of 1.5 hours, and they are not all aspects covered. Contact us, please, if you are more interested in all the details. But what I like to point out is at this place, our new machine AS200Jet Pro and Pharma, which are not only sieving machines. And you see that also good reference in the pharmaceutical application. Many thanks again to Bayer, which allowed us to use this reference. They really like the machine, and for sure others of those voices are following. We launched the machine in October last year, and now we have sold already several to the not only but also to the pharmaceutical industry, and we are really proud of this machine because it is really not only a sieving machine, it is an all-in-one machine which combines sieving, weighing, and evaluation in only one device. We have here all parts which are contacting the sample pharma materials, so the nozzle, sieve chamber, hose, and cyclone, for example. And the very important thing is it has an integrated balance. And it has a reading accuracy of 0.01 gram and calibration accuracy of 0.1 gram. So you can sieve and weigh. And also here with its touch display and the integrated computer inside, you can evaluate everything. Yeah, you have a guided sieving step by step. You do not forget any step and need to start from the beginning on, so it saves time. And you can see the results in graphs and tables. You can work with sieve stocks and so on. So many, many functions here allowed by this integrated computer and the touch display. So that I think it is everything done to reduce human errors, which is very important in giving the best and the most reliable results, which is very important for pharmaceutical applications. And yeah, so here we also offer two versions. The Jet Pro is more or less with the sophisticated basic, let's say it like this, features, and the Jet Pharma version additionally gives some features for working in a GMP-conform way, user management, audit trail, electronic signature, and so on. Yeah, and you can also scan this here to come to the product page later on. So just a short video. I can see some of the aspects of the machine here. The rear side, where you can also attach the vacuum cleaner if you don't use the cyclone, for example. Yeah. And when you use the cyclone, then the sample is collected in this bottle you have seen. Have the touch display, different ways of methods you can use. Fast sieving, like a sieve cut, where you have to calculate everything manually. You can do it, or you get different results in the manual sieving or in the method. In a method, you can select all different sieves you have pre-imported to the machine, give a method name and all the details which are important for the specific method, for example. And you can also not work only with sieve cuts, but also you can use air-jet machine for true particle size distribution with different sieves one after each other. And here you see also how, the balance is working. Yeah. So now it is tiered and set to 0, for example. You also see that you have the different steps in the sieving process. It says you now fill in a sample, put on the sieve, put on the lid, and so on. So you do not forget any step. And it's really nice and easy, especially if maybe your lab stuff changes from time to time. You need to assure that the results are always the same and secure. Like also to come to here, you see how the nozzle moves and the sample is blown against the lid. Agglomerates are broken up and passing through the sieve, for example, and can be also collected in the cyclone. Okay, and you see the progress of the machine. I do not like to show everything right here, but you get different results. Also, you can compare the results of different sieving. You have different checks and balances in order to avoid human mistakes. Yeah. Okay, I think I can skip this. You will get this video also in the download package. So please make sure of the download package in order to get all the information. Here again, the cyclone, which helps to use the fine fraction if you like. Some screenshots again, with the sieving progress here, the results, and the overlay results of different sieving processes. And the clever assistants, so we have many of them, and all of them are designed for as precise results as you can get and avoid human mistakes. For example, the guided sieving, as I showed you before, but there's also, for example, a weighing assistant and a weighing tolerance, which means you put as much sample material as in the ISO standard is allowed for this kind of sieve. So it gives you a hint for that. And you can also use it in your own way, like you always like to weigh in 10 grams with a certain, let's say, a deviation allowed. Then it gives you a warning if too much or too not enough material is weighed in. Yeah. So if there was a mistake, it warns you. We have the back weighing tolerance, which I like personally very much because you expect a specific overgrain after the sieving process. You know, it must be always, let's say, 2 grams. And if it is not 2 grams, but too much or too less, it gives you also a warning. And this gives you the chance to see, oh, stop, there's something with my process, my production process. I need to stop that batch because the particles produced are too large or not large enough, for example. Yeah. So many options here. And also for your qualification process, for the validation, we help with an IQ/OQ risk analysis, which is optionally available. Okay, so this was really a quick overview about many different topics to pharmaceutical application. I think maybe more webinars are following during the next time, which deals more with mechanochemistry or biotech or quality control or whatever. So there will be also, yeah, let's say a survey. Please make use of it. And I think there you can take what are your interests to see what maybe is the next webinar about. And I hope you will also go to our landing pages, pharma, biotech, mechanochemistry, and get more impressions. What is yeah, really done outside in the world with all the different scientists and labs we collaborate and get a better picture of all the details. Okay, so now it's more or less time to look to my colleague. He was busy with chat and the functions and answering. He says all the questions have been answered. Yes. Maybe there's one question regarding the difference between the new sieving machine, AS200Jet Pharma and AS200Jet Pro. So there are 2 different models, so you cannot upgrade them into each other. So you have to decide from beginning on which one you need. And the full version, so the pharma version, gives you this audit trail, GMP yeah, functions, e-signature, user management. And once you activate it, you cannot use it without signing in. So it always needs to know who is working with it, when, and what is done. So you can also not delete any results. Yeah. The Pro version simply has all the functions without this user management and e-signature audit trail stuff. Yeah. So in principle, they are very similar to each other, but there are some more extra functions in the pharma version. Okay. Then there was also a question about fatty sample material. Again, jumping back to the quality control means when you have to crush fatty material, if liquid nitrogen and brittling also helps, yes, it also helps, but it is a little bit trickier maybe than when you have sticky materials. Fatty materials, you can also, of course, brittle the fat. Yeah, and then it is also not that sticky anymore and leading to this smashy, smeary material. What you can also do is working with talc or talcum. When you mix the sample with talcum and then put it into the ultracentrifugal mill, then you have the particles separated because the fat is absorbed. This can also help. Or using distance sieves, for example, yeah, so that you do not warm it that much and you do not squeeze out the fat of the particle that much because you reduce the shearing effects in the machine. So there are some tricks also to reduce the stickiness of fatty particles as well. Okay, then I think we stay in the chat, but we finish the presentation. Thank you very much for listening, and please, you will receive the email with the download package. Do not hesitate to take all the different materials for your information. Okay, many thanks and have a good time. Bye-bye.